Hello, everyone, and welcome to another episode of Dr. Gilbert Hosts. Today, we have the privilege of talking to three APDA-funded researchers, and we're going to hear about what they're working on to advance the field of Parkinson's research. So glad you can join me in watching and listening to these experts in Parkinson's research. Now, these three researchers each were awarded a new type of APDA grant. And this is a grant we're calling the Bridge Funding Award. And it's a grant we have awarded recently for the very first time. And this was in response to a difficult funding climate for Parkinson's disease research. And people were having difficulty obtaining money to do their work. So APDA launched this new award with the goal of sustaining promising investigators who are facing unexpected gaps in their support. They may have received funding in the past, but they've lost their funding or their funding ended, and they have yet to secure the next round of funding. And we didn't want their work to be interrupted. And so we intervened and awarded these grants to ensure that critical research would continue without interruption. So during this broadcast, you'll hear from our three inaugural Bridge Funding awardees, and we will explore how APDA helped keep their Parkinson's research moving forward. We're going to start with Dr. Hwang, and she'll discuss her work on cerebral spinal fluid biomarkers for Parkinson's disease. Then we will hear from Dr. Francino Porciuncula, who will discuss his work on strategies to improve the walking of people with Parkinson's disease. And then third, we'll hear from Dr. Donghae Yang, who will discuss his work on generating dopamine neurons from stem cells. So to begin, I'm going to give a formal introduction to our three special guests. We have Dr. Hwang, and she's a movement disorders neurologist and instructor at Washington University School of Medicine in St. Louis, Missouri. She obtained her bachelor's degrees in psychology and bioengineering from the University of California, San Diego. And following this, she matriculated at the MD-PhD program at the University of Illinois in Urbana, Illinois. She studied telomere biology under the mentorship of Dr. Sua Hwang. Dr. Hwang then completed her neurology and movement disorders training at Washington University, and during her fellowship became interested in biomarkers and therapeutics for Parkinson's disease, and she's currently underway in identification of cerebral spinal fluid biomarkers for Parkinson's. Welcome, Dr. Hwang. Next up, we have Dr. Francino Porciuncula, who's a research assistant professor at the Sargent College of Health and Rehabilitation Sciences at Boston University. She's trained as a physical therapist and rehabilitation scientist. Dr. Porciuncula's research focuses on developing innovative gait rehab interventions in Parkinson's by targeting biomechanical, cognitive, and behavioral processes that underlie gait dysfunction. His research seeks to develop and leverage next-generation wearable technologies, including sensors, robotics, and digital health solutions to improve gait outcomes. Dr. Porciuncula received his doctorate in motor learning and control from Columbia University and completed a postdoctoral fellowship in clinical biomechanics at Harvard University. Welcome, Dr. Porciuncula. And finally, we have Dr. Donghae Yang, who's a postdoctoral fellow in Dr. Lorenz Studer's lab at Memorial Sloan Kettering Cancer Center in New York. His research focuses on leveraging cutting-edge genetic, human pluripotent stem cell technology to understand, model, and potentially treat neurodegenerative diseases such as Parkinson's. Dr. Yang received his PhD in 2022 from the University of Toronto in Canada, and currently, his research aims to dissect the development of midbrain dopaminergic neurons with a particular focus on the A9 subtype, which is responsible for motor control and is most susceptible to degeneration in Parkinson's. The goal is to generate A9 midbrain dopaminergic neurons, both to model Parkinson's in the lab and then to develop novel cell replacement therapies for Parkinson's. Welcome, Dr. Yang. We will now start our presentations. We'll listen to one from each of our guests, and then we'll all come back on camera for the Q&A. So get your questions ready. Now, please be aware that the work that will be presented today is new and therefore not published yet. So please do not take screenshots of anything you see today. So let's start with Dr. Hwang. Dr. Hwang, welcome to our program and take it away.
Thank you so much for having me, Dr. Gilbert. So today I will be talking to you about my work that I've been doing with this bridge funding. My title of my talk is Potential CSF Biomarkers of Parkinson's Disease Progression. And I'm going to slowly take you through a little bit of a background. As you may know, this is the audience that knows that Parkinson's disease affects about 1 million people in the United States. And currently, it is purely a clinical diagnosis, meaning a neurologist or a movement disorder specialist spends time with the patient and he or she watches you move and have you do a whole bunch of movement maneuvers and then determine whether or not you have Parkinson's disease or not. While the movement disorder neurologist, is quite good at this, typically 90% good at diagnosing Parkinson's disease. Often they're not as good as diagnosing or separating out the mimics, the Parkinson's disease mimics. There are motor features in Parkinson's disease that we all know: tremor, slowness, stiffness, balance and gait issues. And there is also a wide range of non-motor features like lightheadedness with standing, orthostatic hypotension, depression, anxiety, fatigue, REM, sleep behavior disorders, and cognitive impairment and dementia. However, to date, there's no biomarker, meaning there's no lab test or no imaging study that tracks this disease, and therefore there hasn't been a good disease-modifying therapy. So some background of Parkinson's disease. We think the defining feature of Parkinson's disease is due to a protein named alpha-synuclein. And this protein, when it exists as a monomer individually, is crucial to normal neurological functioning. You, me, everyone has normal alpha-synuclein. But when this protein becomes aggregated or accumulates with other alpha-synucleins, it can become deposits, and it can deposit into cells, and we call these neurons, neurites and neurobodies. And often when you have too many of these deposits, it causes neurodegeneration. There is also this hypothesis or this thought that as disease progresses, often starting first with constipation or differences in pain, that it progresses from the peripheral and into the back of your brain, into the midbrain, and then fully into the cortex, the neurocortical region. So there is this Barak hypothesis of progression that we see with this deposits of alpha-synuclein. However, right now, the gold standard for diagnosing Parkinson's disease is that brain autopsy, and then that's not good. We don't want that for you or for anybody. And what we really need is a lab test or imaging test to be able to diagnose Parkinson's disease better. So therefore, there's also a really, so currently there are a few tests out there that can try to help with diagnostics of Parkinson's disease. There's a CSF alpha-synuclein fibroamplification assay out there. However, these assays are not quantitative, and they cannot give you any information about how far you've progressed with your disease, or it cannot give you any information about how fast you will progress in the future. There's also, you can compare this alpha-synuclein, the total alpha-synuclein, not just of the aggregates, but with everything, like the normal and the abnormal in your CSF. But that seems to be, while it is lower in people with Parkinson's disease, there's a much overlap between normal participants and people with Parkinson's disease. So that's not a really good marker of how bad your Parkinson's disease is. And so there is a really big need in the field to find biomarkers or a lab test that's able to find cures or disease-modifying therapies. These biomarkers can, one, improve the diagnosis. Like you can say, oh, you run this lab test, you have Parkinson's, you do not. You have a mimic, you do not. You have Parkinson's or you do not. It can actually also help with target engagements of potential drugs. So for many clinical studies, I feel like the clinical trials in the field haven't worked well because we are not recruiting from, we're probably recruiting for people with Parkinson's disease and people with the mimics as well. So we're not very good at the recruitment. And so if we have a good way to diagnose, a better way of diagnosing Parkinson's disease, we will probably be able to recruit better for the clinical trials. And the last point is that biomarkers can track the progression of the disease better. So maybe you can say, hey, this there's like a surrogate for how much neurodegeneration or how much cognitive impairment you have based on this lab marker. So I have been very lucky. I am at Washington University, and we have the largest single institutional longitudinal study in Parkinson's disease. And this is a collaborative effort between multiple labs on campus. It's Paul Kotzbauer, Rick Campbell-Perrin, Megan Campbell, and Joel Perlmutter. And together, there's 2 longitudinal studies, and we have probably more than 450 participants currently. And these participants are both Parkinson's disease at any level of, we recruit them at any level of disease or any stage of disease, and then also with control participants. And then every 2 to 3 years, they get extensive cognitive testing, motor testing, and psychiatric assessments as well. And every 2 to 3 years, they also donate CSF, blood, plasma, and get MRI scans and PET scans. And at the end, they have agreed to donate their brains for research. So we are very thankful for these participants. And the goal of this study, these studies, is to help identify some biomarkers for disease identification of Parkinson's and its mimics, and for the progression of disease. So the main hypothesis is that I expect whatever we see, the changes in CSF proteins will reflect the changes we see in brain. And so and these participants are very generous. They have most of them, like more than 50% of them, have donated CSF, and often it's like 30 mils of CSF every 2 to 3 years. So we have many. And so the method of my study is I have compared brain tissues using SomaScan, which is a scan that which identifies the relative levels of more than 7,000 proteins in PD participants between the different cognitive statuses: normal cognition, mild cognitive impairment, and then dementia. And so in that case, I can see which proteins are upregulated and which ones are downregulated. And I can compare that in brain tissue and that in CSF as well. And the ultimate goal, well, after that, I plan to validate 2 or 3 of my top candidates from CSF to make sure these are the ones that are changing and are good surrogates for cognitive decline. Ultimately, though, my goal is to identify a panel of CSF proteins that can provide a biomarker for the progression of cognitive impairment in PD. I think this is the initial goal, and of course, future goals can be to reflect motor symptoms and psychiatric symptoms, but I think currently I might focus on cognitive symptoms in Parkinson's disease. So this is just a graph of preliminary data. This is brain tissue proteomic data comparing PD with dementia and PD with no dementia. And as you can see, we applied a very strict criteria, much higher than like the 0.05, because there are so many proteins. So we applied a very strict criteria. And what we see is there's 4, 5, 6 proteins that come up that are significantly different and change. Actually, they're significantly lower in people with dementia compared to people without PD participants without dementia. And so based on this, I'm trying to develop CSF immunoassays, single immunoassays, to try and validate these candidates. So that's a little bit overview of my research so far. I think I want to thank APDA so much for this bridge funding. I had my funding interrupted previously, and this is what the bridge funding is keeping my project going. These are also mentors and PIs that I'm working with here at WashU that are involved in the longitudinal study in PD. And then, of course, I'm very thankful for the people with PD and controls that participate in our studies. Now, without further ado, there will be questions at the end, but I want to introduce Dr. Porciuncula, and I will pass the baton to him.
Thank you, Dr. Hwang. I'm very excited to be sharing with you today my research here at Boston University. But first of all, a big thank you to the APDA for supporting our study to be able to do this important work. It's a big privilege. Here at Boston University, we're interested in examining and developing practical solutions to improve walking in Parkinson's disease. And one promising approach is through the use of rhythmic auditory cueing. This is walking and listening to a simple metronome beat or listening to music. And we're examining how that helps with the brain and body in coordinating movement to help walking become less cognitively demanding, less effortful, and more automatic. Improving walking in Parkinson's disease is a key goal that we work on in rehabilitation because we know its impact on quality of life. Walking is primarily an automatic task. It's second nature to us. We don't think about our walking. And this is the exact control that allows us to do multiple things all at once without feeling unstable on our feet. But in Parkinson's, this disruption in the circuitry in the basal ganglia results in this loss of walking automaticity, leading someone to, I'm just going to advance my animation here, leading someone to use other circuitry that is more attention-based. And this essentially leads to having someone think about their walking. So when someone thinks about their walking, for someone who has Parkinson's, they can make it better. They can take larger steps. They can walk faster. But they have to think about every step. And that's not realistic and it's not sustainable. It is mentally tiring to do that type of strategy. And ultimately, it limits overall daily mobility. So we still are very much in need of effective solutions that can improve upon this issue of automaticity of walking in Parkinson. One promising approach, as I have mentioned, is rhythmic auditory cueing. This is a rehabilitation strategy that pairs rhythmic cues with walking, whether that's listening to a simple beat of a metronome or listening to music. And what studies have shown, this tends to improve walking. So it makes someone walk with bigger steps, faster walking speeds. What's also exciting about this approach is that it taps onto alternate circuitries in a brain, bypassing the impaired basal ganglia. Rhythmic auditory cueing is mostly used in the clinic in physical therapy, but it has a lot of potential to be used in daily life in people's homes to promote a more routine walking regimen that we know is essential in the long-term management of Parkinson's. But the success of this kind of like an expanded approach really hinges on whether this rhythmic auditory cueing reduces cognitive demands of walking. Does this actually promote automaticity? We don't know quite just yet because of just the limitations in our methodologies over the past several decades. Our understanding of walking has primarily relied on MRI studies, fMRI, and this is essentially done lying still in a capsule and not really capturing actual walking. Over the past several years, there's been exciting advancements in mobile brain imaging through this technology that's called FNIRS, which stands for Functional Near-Infrared Spectroscopy. This is a non-invasive, portable mobile brain imaging technique that uses infrared light to measure or to estimate brain activity when someone's doing typical daily activities such as walking. And this is the technology that we've used in this study. To date, we have enrolled 30 participants with Parkinson's, and we've had them use this FNIRS system. And this comprises of wearing a lightweight cap, which secures our sensor setup of light sensors. And these sensors are placed on the region of the brain that corresponds to the prefrontal cortex. This is the region that is responsible mainly for attention. And this particular region tends to be overactive in Parkinson's. It is more active than usual. And this is indicative of this impaired automaticity of walking. Alongside FNIRS, we also use wearable sensors to capture the quality of their walking. And we had participants walk up and down the hallway performing different tasks, including regular walking, walking and counting, that is subtracting backwards by threes, which is supposed to be an attention-demanding task, walking with the beat of a metronome set at their usual tempo and at a slightly faster tempo. And these conditions were randomly administered across a 10-minute bout of walking and repeated 3 times. What we're measuring here for FNIRS is the change in oxygenated hemoglobin in that part of the brain that is responsible for attention-based walking. And this is in respect to regular walking. So this is what we found. So this is a graph of brain activity across the 3 conditions. And I'm going to draw your attention here onto the green and onto the orange, which corresponds to walking with the metronome beat set at usual tempo and fast tempo. And we see reductions in brain activity. This indicates that there is less attention needed during walking with cues. In contrast, we see an increase in brain activity when someone is walking and counting, not so surprising because that is much more distracting and attentionally demanding. These results are providing some strong evidence that when walking with rhythmic cueing, this reduces activity in the prefrontal cortex, which is not necessarily needed during walking. Therefore, this may be a signal that this is improving one's walking automaticity. We then looked at what was happening to their walking. So we measured walking quality based on the length of their steps, which is stride length, and how fast they were walking, which is walking speed. And what we are seeing here is that with walking with cues, either at usual tempo or fast tempo, people were walking with bigger steps, as you would see here in stride length. And it has corresponding effects with faster walking at usual tempo and even faster walking when you speed up the tempo a little bit more. In contrast, we see decline in walking quality when someone is distracted, walking and counting, which is not so surprising. Together, these results in brain activity and walking quality are really exciting because they have corresponding benefits for such a simple approach of listening to a rhythmic cue. So the key takeaways from our study thus far is that rhythmic cueing reduces cognitive attentional processing with improvements in walking quality. And for this relatively simple procedure, these are immediate changes without any training. Delivering rhythmic cueing tends to augment walking, and it has immediate improvements in walking automaticity and Parkinson. So we're really excited about this. What the APDA Bridge Funding Study is allowing us to do is to further expand on our examination beyond metronome cues. Metronome cues is kind of like the first stage in our examination, keeping the protocol very clean. But as we know, people would gravitate more towards something that's more enjoyable, such as music. So in the next round of our data collection, we are examining what happens to brain activity when someone's listening to music. And in parallel, we're also exploring other technologies that are currently available with partnerships with industry partners to see what that effect is on one's walking ability and their walking automaticity. And with that, I'd like to thank our research team at Boston University for their significant contributions, and once again to the APDA for their funding support. Looking forward to your questions at the end of our talks. I'm going to pass the baton now to Dr. Yang.
Thank you, Franchino. Good afternoon, everyone. My name is Donghei Yan. I'm a postdoc in the Sloan Kettering Institute in New York. It's a real privilege for me to be here today, and I'd like to share the work that we're doing in the lab to make dopamine-producing neurons from human stem cells. And we actually use these cells to better understand Parkinson's disease in the petri dish, and also to help move toward new treatment strategies, which I'll highlight later. So to start my presentation, I just want to introduce one key idea, which is that not all dopaminergic neurons are the same. There are different groups of dopamine-producing neurons in the midbrain that I highlight here. And the 2 important ones are called A9, that I label in orange, and A10 in green. And A9 neurons, they're found in this brain region called substantia nigra. This is very pigmented region of the brain. And these are the cells that are especially important for movement controls, and they're also the ones mostly affected in Parkinson's disease. Therefore, their loss contributes to symptoms like tremor, stiffness, and slowness, as you can imagine. Whereas A10 neurons, they're nearby, but they play somewhat different roles. They include motivation control, mood, and reward, as well as cognition. So even though these two groups of neurons both produce dopamine and they're both located in midbrain, but they're not really interchangeable. So this one major question in Parkinson's research is that why A9 neurons are more vulnerable while A10 neurons are more resistant in the disease. And that question really matters because if we want to understand Parkinson's disease and eventually replace the lost neurons, we need to focus on the right type of neurons that are lost, which is A9 in this case. So how do we make dopaminergic neurons from human stem cells in general? Right. So we start with this type of stem cell called human pluripotent stem cells. And this is type cells can virtually become any cell type you can find in human body. So here I am showing you, we guide these stem cells step by step into becoming midbrain dopamine producing neurons by giving them instructions, like biological instructions that mimic normal development. And by day 16 in the petri dish, these stem cells will become young dopaminergic neuron precursors, or what we call them progenitors, and later they will mature into dopamine-producing neurons. And this approach has opened up many exciting avenues of research. One is that it gives us way to study Parkinson's disease using human stem cell-derived neurons in the lab. And another very important application is cell replacement therapy, where these human PSC-derived cells may one day help restore lost function in human. And our field has already made important progress, as I highlighted here, with these three papers. And I also want to highlight that there are early clinical studies publishing last year, 2025, have shown very encouraging signs. But there are still challenges we need to solve. And one major challenge is that the current methods often produce a mixture of cell populations. So even if we make dopaminergic neurons from human stem cells, we do not always have control over exactly which subtype or groups of neurons we're getting. And for Parkinson's disease, this really matters because the main cells that are lost are A9, as I mentioned before. And when people study stem cell-derived grafts in animal models, as I'm showing here with this study, we always find a mix of cell populations. So each cell population is encoded by different color. As you can see, this graph contains not only the A9 neurons that we want, it has a lot of contaminating populations. And for therapy, this is not ideal. And also for research, it also makes it harder to study the most vulnerable cell types, which is A9. So this becomes the central focus of my postdoctoral work, which is can we move beyond making general dopaminergic neurons and instead learn how to make the right subpopulations in a controlled way. And I am happy to tell you today that we're now able to generate both A9 and A10 dopaminergic neurons from human pluripotent stem cells. For today's talk, I'm not going to go into all the technical details, but in short, we used stem cell biology together with computational and genetic analysis to identify essential signaling events that help guide cells towards one subpopulation or the other. So here I am showing you some pictures. The top cells are A9 neurons, and the cells on the bottom are A9 neurons. Oh, sorry, top are A10 and bottom are A9. And importantly, these neurons show differences we would expect. For instance, A9 neurons show more of a marker called ALDH1A1, highlighted in red, whereas A10 neurons show more of another marker, CLB1 combined in gray, as I highlighted here. So this analysis really gives us confidence that we're actually making distinct dopaminergic neuron subtypes in a lab in the petri dish. And on top of that, we also wanted to know whether these cells behave or function differently, not just look different, right? First, we measured dopamine synthesis ability in these neurons, and we found that the A9 neurons stored and released much more dopamine compared to A10. And we also look at their ability of making pigments. So as I mentioned, a hallmark of neurons in the substantia nigra is that they will gradually develop dark pigmentation. And strikingly, as you can see in this picture, only the A9 neurons develop a visible pigmentation in our culture, while the A10 neurons mostly did not. So put everything together, these findings suggest that the A9 neurons we're making in the lab from human stem cells are beginning to resemble the real A9 neurons that are especially vulnerable in PD. And this also gives us the opportunity to ask the next very important question, which is, can these human PSC-derived A9, A10 mature neurons survive after transplantation in animal brains? And before I continue, I just want to shout out to APDA and of course the donors. This part of work has been supported in part by the Bridge Fund Award. So we're carrying out these preclinical animal studies in collaboration with Dr. Vivian Tabar's group here at Sloan Kettering. And together we're testing whether purified, more mature A9, A10 neurons, whether or not they can survive and integrate after transplantation into animal stridums. And of course, also substantia nigra. And this is still ongoing work. It is technically challenging, I have to say, but I just want to share with you some encouraging early data. In this picture, what I'm showing is that when we transplanted A9 neurons derived from human PSCs into the stridum of a mouse, we found that these human PSC-derived neurons were able to survive and also integrate into the animal brain. And this is an important first step and really give us motivations and hope to keep moving forward. And finally, I just want to thank many people who made this work possible. Of course, my mentor, Dr. Lawrence Duder, for guidance. And I'm also very grateful to our collaborators, Dr. David Seltzer at Columbia, and also Vivian Tabar at Sloan Kettering. Also want to shout out to APDA to supporting this work through both a postdoctoral fellowship and Bridge Fund Award. And thank you all for listening. I guess it's time to invite the other speakers and Dr. Gilbert back for Q&A. Thank you.
Yes. Thank you. Thanks to all our speakers. What I was thinking when I was listening to all these talks is how each talk is so very different and really approaching Parkinson's disease and Parkinson's disease therapeutics from three entirely different perspectives. And I think that's really wonderful because it takes lots of different ideas and lots of different brains to get to the solution. So thank you all for your wonderful presentations. We have a lot of questions that have come in already. And so what I'll try to do is focus on the three different projects. We'll start with Dr. Hwang's project. And I'll try to group some of these questions together. So there were a couple of questions about, you mentioned mimics of Parkinson's disease. So could you discuss some examples of those mimics and how does having a Datscan relate to a diagnosis of Parkinson's and potentially how a Datscan would be viewed as a distinguisher between a mimic and Parkinson's disease and how a Datscan folds into this biomarker discussion. So Dr. Hwang.
Sure. So I mean, I use the mimics term pretty broadly, but the mimics, Parkinson's mimics, you can think of, there's something called Parkinson's plus syndromes. These typically include diseases called multiple system atrophy, progressive supranuclear palsy, corticobasal syndrome. A lot of them have Parkinson's features, but they also have other features as well. And neurologists and movement specialists are not very good at diagnosing these mimics, so these Parkinson's plus syndromes. I think if we compare it to autopsy study for PSP, progressive supranuclear palsy, we're only about 50 to 70% accurate when we're diagnosing them. They're often one of the others, or idiopathic Parkinson's disease, which is the most common. However, that being said, idiopathic Parkinson's disease is very common. It's most likely neurologists are pretty good at diagnosing that. I think the sensitivity is like 95 to 97%, depending on which literature you look at, and just based on clinical symptoms. That being said, the Datscan is sometimes used. And the literature, I think, for the Datscan, it's very dependent on the slices, the imaging study that you get, and also it's about as good, the way I was trained was, it's about as good as the neurologists. And so we at WashU don't routinely order the Datscan to help with the diagnosis of Parkinson's disease. That being said, the Datscan could also be positive for some of these Parkinson's plus syndromes. So it's not that great at separating the mimics from Parkinson's disease.
Great, thank you so
Bye.
much. I'm sorry, yes, I think that answers a few of the questions that were posed. Thank you. And we have a question from Alan, which is the Wash University study still taking new applicants? And can a person not be in the St. Louis area
Yeah, that's a
to
great
participate?
question. That's great to be interested. So we are taking new participants all the time. The key is you have to be able to come in for some of these cognitive motor psychiatric imaging studies every 2 to 3 years. These studies usually we take people locally, basically who are established here in the St. Louis, not necessarily with WashU movement, but anyone in the area that's interested. And so I do think if you're interested, you can participate, but you do have to be able to make trips to St. Louis every 2 to 3 years and agree to brain donation at the end of your life.
Thank you. And final question for Dr. Hwang before we move on to Dr. Porciuncula's questions is, are you studying other things besides cognitive decline?
Yeah, so I think right now, initially, we're doing cognitive decline because that's the most well characterized in our data set. We have like a large battery of tests to measure cognition. That being said, you could easily apply the same methods, the proteomic and the immunoassay developed method, to motor psychiatric symptoms or any other symptoms like freezing of gait that you may have, like you can find.
Excellent. So the method is there. You're starting with the cognitive, and it can be applied to other things. So that's absolutely wonderful. Let's move on to some questions for Dr. Porciuncula. We have a great question from John, who's asking, can you use this FNIRS technology on someone who has DBS in place?
The answer is yes. This is what's exciting about this technology. It's non-invasive. It uses light. There would be some considerations on how we analyze the signals, especially for the light sensors that line up where the DBS is. But DBS is not a contraindication to using FNIRS. So it really opens up our participant profiles to understand different presentations, even those with DBS.
Excellent. So we have a few questions. We have Michael, Victoria, Jonathan. They're all asking about sort of the actual specifics of how this works. What is the tempo, for example, of your rhythmic cueing? How would rhythmic cueing be used in everyday practice? You mentioned maybe music could be a useful tool instead of rhythmic cueing. Are there specific types of music that would be useful? How is this music delivered? Is it through headphones? Can you give sort of a nuts and bolts of what it would look like to be either using rhythmic cueing or using music to help with the walking?
Yeah, these are all fantastic questions. So I'll start with a tempo. So we usually start with the person's baseline tempo. So we usually measure one's cadence or number of steps per minute at their usual comfortable walking speed. So that's kind of like what we regard as 100%. And then that's kind of like our starting point. And then our goal, well, the goal in gait rehabilitation in Parkinson is to have someone walk a little bit faster to get a little bit more intensity in their walking. So we usually modulate that, upregulate that by maybe another 5% or 10%. So that's typically the band from 100% to 110 or maybe 115%. But that also depends on where a person is starting. We don't want to make someone walk even faster when they are at their high walking speed range, when they're achieving that range. So it's highly individualized. So it's really beneficial to work with a physical therapist to kind of like determine the preliminary parameters. Now with music, we recommend having users listen to music that they enjoy because that's what's going to keep them engaged. The type of music is more on, I think the main parameters in music is more on the frequency, the steps or the beats per minute, and how salient these beats are. So, for example, listening to strings like an orchestra would be harder compared to something like oldies, right? When you have a very tangible and specific beat. So that's another thing to consider. I forget. So another question about headphones. I guess from our protocol, with our protocol, we did it with just playing off an electronic device. But in our other clinical trial, we have had participants listen to music through bone conducting headphones because this was kind of like being done out in the real world, and we wanted them to have situational awareness of what's happening in their environment and not pose a safety risk. So it's highly context specific and individual specific.
Fantastic. Thank you very much. So let's go to Dr. Yang. We have a good question from Michael, which is, what do you think is the most optimistic time frame for when stem cell research will result in treatments for people with PD?
Time frame in terms of like policy making or treatment outcome?
Yeah, when you think this can be a treatment available to people with Parkinson's, a stem cell treatment.
Well, since I'm not a clinician, I may not have the most comprehensive information, but what I can tell you is that so we have clinical phase 1 and 2 being completed in Asia, specifically Japan and North America and Canada and United States. And I think the company is recruiting patients for clinical phase trial 3. It's ongoing. I don't know when they're actually going to prove it. So I can't really give an answer for that question, but in terms of treatment, I think by 18 months in both clinical trials, they see a significant improvement in, I think, the off score.
So it turns out that there is the very first stem cell treatment was approved in Japan a couple weeks ago.
Yeah.
And it was based on a very limited amount of data that probably wouldn't be enough to have it approved in the United States. They have a kind of a special approval track in Japan that kind of allows more limited data to lead to approval, with the caveat that more data will be collected. And so there is a stem cell treatment approved in Japan right now. So that's exciting, and it means that work of Dr. Yang's is really something that's very, very relevant to try to find really the best way of introducing this type of treatment into a human brain. So this is really exciting and very relevant work. And then in terms of Joanne's question, are there clinical trials? So the company, as Dr. Yang mentioned, the company that was working both in America and Japan on this type of treatment has a trial going on in America for a similar treatment to the one that's now available in Japan. That information's on our website. If you go to our website and click on the article about the approval, the Japanese approval, you'll find information there on the clinical trial that is open in the United States. So that's very exciting. So I have a great question from Gail for Dr. Yang is, do the A9 cell, theoretically, if you produce A9 cells and you inject them into somebody with Parkinson's, will those A9 cells get Parkinson's themselves after a while? What's your hypothesis about that?
Yeah, that's a really good question. So the approach we take is called genetics and cell therapy. So genetics meaning that when we get patient iPSCs that carries mutations, for example, LRRK mutation, alpha-synuclein mutation, we'll correct them back to wild type first, right? And that will already kind of reduce the risk of them regaining Parkinson's disease. And we'll use these healthy stem cells to generate A9 neurons. So I guess in that way, we can already like have a lower risk of them being primed to develop the disease. And so that's one way I would answer it. But A9 cells, as you can imagine, they're very vulnerable. They're very like susceptible to all kind of like alpha-synuclein aggregation, as Dr. Hwang mentioned, and also like mitochondrial stress, metabolism stress, all sorts of stress, right? But I would say they might, but we do not have any evidence to claim anything yet, because we just simply haven't done the work. But I can confirm that in animal, especially in mouse, after 8 months after transplanting these human PSC-derived A9 neurons in the striatum, even with some toxins, they do not really degenerate and they do not have Parkinson defects.
Fantastic. That's really good news. We have a question for Dr. Porciuncula from Janet. Are you taking more study participants? What are your requirements to participate?
Yes, so our study's ongoing. We are mainly looking, well, participants should be able to make a visit here at Boston University for this particular study. We are looking for participants who are able to walk at least 6 minutes of walking unassisted. So pretty broad inclusion criteria for this particular study. But we have
Excellent.
ongoing studies that we're going to be doing in the fall for a longer clinical trial intervention based. So stay tuned on that project.
Fantastic. I now have a question that I want to pose to all of you, which is what is the funding climate like now in Parkinson's research? And what do you think will be the obstacles when you need to go search for additional funding? What are your thoughts on that? So maybe we'll start with Dr. Hwang.
I think the funding situation is still pretty dire currently. I think this is a trend. Like there's a lot of young investigators here at WashU have been leaving the field because funding situation is so dire. But I think this it probably will shift. More people are looking for private foundation funding instead of the NIH and government funding, and hopefully it's just like a pendulum and it'll swing the other way very soon.
Dr. Porciuncula, what's your experience been?
Yeah, I think the same here at Boston University. I think the institution has done a great job trying to kind of like handle these changes as they come. But we definitely feel all over the institution changes in procedures, introducing challenges in how we do our work. And then in addition to that, kind of like thinking of like the next funding cycle, what's going to happen after this. I think there's a greater competition for funding support, and being able to produce robust data, I think is really critical in being successful on that next step, whether it's federal or private foundation.
That makes a lot of sense. Dr. Yang, what are your thoughts on this?
Well, since I'm still a postdoc, I don't have experience applying for R01s, but what I've heard that the possibility of securing R01 is just like drastically reduced compared to previous years, from I think from 30% to maybe 20%-ish published this year. So that's not a promising stats. And also our lab are looking for private fundings as well from Michael J. Fox Foundation, from you guys, from Parkinson's Foundation, and hopefully we can transition through this difficult phase, and hopefully NIH will be back to normal.
Excellent. So really all three of you have had very similar experiences. So, foundations, as you said, have tried to step up to help out as things unfold. So we're happy to be supporting you. I wanted to ask all three of you another question, which is if you solve the current problem that you're working on, so as the presentations were presented, the wonderful work that you're doing, let's say you've solved them, what would be the next thing that you would like to do as follow-on from the work that you're doing now? So let's start with Dr. Hwang. What would be kind of the next step in your research?
So like the ultimate goal from for my research project is it's a CSF panel for disease progression, right? So it could be cognitive symptoms, motor symptoms, psychiatric symptoms, freezing of gait. However, if we do get that, or I think a serum or blood test is even more easily accessible than CSF and asking people to give CSF samples in general. It's just much less invasive and much easier to get from people. So, I mean, ultimately,
Excellent.
I think a lab test is my goal.
Great. So you would take someone's blood and you would be able to assess what symptoms they have, what symptoms they might develop, and might be a whole panel about what their particular Parkinson's would potentially look like. Be very exciting. So Dr. Porciuncula, what's the next step in your work?
Yeah, the next step for us is to really examine what changes when someone engages in a walking program that leverages rhythmic auditory cueing over months of treatment. What we've looked at so far is just a single session study, and we've seen really exciting results. And we're very curious on whether we would see these effects or these improvements in walking automaticity after training over months, which kind of like mirrors what would be typical in rehabilitation. We want to understand whether walking automaticity is modifiable with the right targeted training in Parkinson.
Great. And Dr. Yang?
Yeah, wait, can you hear? Yeah.
Oh, the question is, what is the next step?
All right.
You've isolated your A9 neurons. They're very pure. They seem to do what they're supposed to. What is the next step for you?
So the next question that we'll pursue is to understand why A9 neurons are more vulnerable than A10 under different type of stress. So we are planning to stress these subtype of dopaminergic neurons with different type of agents, like chemical agents, aging agents, and also different type of mutations such as LRRK, GBA, those common PD mutations, and see how they respond to different type of stresses to really understand the underlying mechanism why they're so vulnerable.
Fantastic. So we have time for one last question, and I'm going to ask this one last question to all three of you as well. And that question is, there's a lot of frustration among the entire PD community about why research is so slow. Why is there no good treatment? I mean, there are lots of good treatments. I shouldn't say there are no good treatments. There are many good treatments, but there isn't a cure, and there isn't a fabulous treatment that sort of takes away all the problems. So people with Parkinson's do still have a lot of challenges. What are the biggest obstacles to really getting over that hurdle and finding that ultimate treatment or cure? Why do you think the research is slow and hasn't happened yet? So let's start with Dr. Hwang.
Of course, I'm biased, right? I think there's no good biomarker in the field, good lab test in the field. So that's really slowing down clinical trials and being able to select for your participants that you want to participate in the clinical trial appropriately so that the clinical trial works. There's no good biomarker for target engagement, so you don't know if your clinical trial drug works. You're asking, I don't know. So I'm very biased. I think part of it is there's no good biomarker. But why is there no good biomarker? I think research in general is just very slow. And so the work that I've shown, a few pieces of preliminary data probably has taken 1.5 years already to get. And so it's probably going to take a few more years to get some preliminary data and then to transition from the preclinical state to a translational state and to clinical trials is going to probably take another 10 years, is my guess. And so I think that is very frustrating to a lot of patients, just how long it takes. But research in general just takes a long time. There's always goods in that when you have great data, it's fabulous, it's very exciting, but a lot of times there's a lot of failed experiments as well.
Fair enough, fair enough. Dr. Porciuncula, what do you think is the biggest obstacle to taking the next big leap in Parkinson's research?
Yeah, in my experience in gait rehabilitation, we recognize that PD is a very heterogeneous group. There are different gait presentations, and we don't seem to do, well, we can do a better job in capturing the range of phenotypes or types of gait presentations. And I think that speaks to making research more accessible and available beyond laboratory settings. So we're starting to think about doing research in people's homes, people's own communities to really understand how people perform in a community and also to reach people who don't have the means to get to labs and universities or academic centers at regular intervals. So I think if we can improve our sampling, understand the population better, I think we're going to be making significant improvements in our research.
Fantastic, thank you. So Dr. Yang, what do you think is the biggest obstacle to why we haven't sort of solved Parkinson's so far?
Well, I think I'm just going to comment from the cell therapy side. So my mentor, Dr. Lawrence Duder, he's been thinking about making A9 probably for 15 years. And that has never come true. But like sporadically, there will be 1 or 2 A9s. And it's not until last year we figured out a robust way to also a controllable way to make A9 neurons. So it really takes a lot of time, and it's really and the reason that came out the strategy that making A9 was because the development of technologies such as spatial transcriptomic and single cell technologies, which are emerging in the past 5 years maybe. Yeah, so it's really, and especially when you think about these are the very, very specific neurons in such small number and this very precise location of brain, you can imagine just how hard it is to make from stem cells. So it's really like the technology just came out and we took advantage of it and found a way to make it. And also another reason that's very slow is because it just takes a lot of preclinical studies to get to that point. For example, once we transplant A9 and 10 neurons, first we have to solve the survival issues of neurons. That's not easy. But after that, we have to do all sorts of behavioral analysis, and that can only be done after 8 months after transplantation. So everything adds up, and then the process just really long.
Yeah. So research takes a long time when it's done well. But as you said, with advancing technologies and the merging of the advancing technologies with good ideas, there is progress being made. So that's a wonderful piece of information. So thank you. I wanted to thank all three of you for your presentations, your fantastic information, and for really demonstrating how important it is for APDA and other organizations to step in and provide funding for young investigators like yourselves to get your projects to the finish line. So thank you so much for helping us explain that to our community. So if you knew someone who missed this program today, and if you joined late, if you want to watch again, this recording will be available very soon, potentially even today, on our YouTube channel. And when you're there, don't forget to subscribe to APDA's YouTube channel, and you will see all our videos and be alerted about any live broadcasts. So again, thank you so much. We've had questions in the chat about how to reach out to our researchers to join their trials. So we're going to collect some contact information and be sure to share it with you on our post-event email. So look out for that. I want you to join me on Wednesday, April 29th for our next broadcast, and that will be just with me, a session called Ask the Doctor Anything. So get your questions ready and be sure to register today for the next broadcast. So for additional information, for resources, please visit our website at apdaparkinson.org. You can please stick around for a few final messages. Before I go, I want to thank all our guests today for being here, for sharing their information, and I want to thank you, our audience, for joining us, and we hope to see you soon on another APDA program. Have a wonderful afternoon, everyone.
I'm Leslie Chambers, President and CEO of the American Parkinson Disease Association. Thank you so much for joining us today on our program. Every day, APDA is committed to providing helpful programs just like this one, as well as a wide variety of support groups, exercise classes, and educational programs to support the Parkinson's disease community. Whether online or in person, APDA is here for you every step of the way. You can find all of our upcoming virtual events on our website at apdaparkinson.org/events. We keep you posted on our social media channels as well, so make sure you follow us there. You can also connect to local resources and events through our nationwide network of chapters and information and referral centers. Remember, we're just a click or a phone call away. If you've enjoyed today's broadcast, I hope you will consider making a donation to help keep programs like this possible. Every gift, large or small, helps APDA support people living with PD through thoughtful programming, reliable resources, groundbreaking research, and more. Please visit apdaparkinson.org/donate today to support our critical work for those with Parkinson's. We are so grateful for your generosity.